Assalamu alaikum everyone. Welcome back to my channel Regal Biology with Khadija. This is another lecture in our NCBI Bioinformatics series. In our previous videos, we started our journey with NCBI. First, we learned about the basics of NCBI, including why this is important, and then we explored NCBI gene database. We learned about what is gene database, how we can use this gene database, how we can download genomic DNA, coding DNA, or messenger RNA. Now, in this video, we will move from gene level to protein level. In today's lecture, we will explore NCBI Protein Database and we will learn how to analyze a complete protein record using a protein example, BRCA1 from Homo sapiens. We will understand that how can we get information of protein record, how to search protein in NCBI, how to understand a protein accession record, how to read protein annotations, and how to identify protein domains and functional regions, how can we understand conserved domains and sequence analysis or alignments, and finally we will explore protein structure information. So without wasting time, let's explore our protein database. Protein record is actually in NCBI, it contains much more information than just an amino acid sequence. After selecting protein in selection menu, we will name a protein. Suppose we want to study BRCA1 protein. Let's search this protein. Now we want to study this protein in Homo sapiens. Here you got a result, the same protein name in different organisms. Like these are, we will select Homo sapiens. Now we will analyze the whole protein record, starting with locus. This locus AAC37594. This is the accession identifier of a protein record. This number is unique identification. It is assigned by this NCBI website to this protein. Next is 1863 amino acids. It indicates protein length. AA means amino acids. Now, the protein BRCA1, it contains 1863 amino acids. Next is linear. This indicates the protein exists in a continuous linear amino acid chain. Next, it is the date. It represents the date of record or updating date of this protein. Before date, there is a word PRI. This is actually primates. It represents the taxonomic division. Here it represents the taxonomic divisions, which is Homo sapiens. Section is the definition. It means the protein name BRCA1 in Homo sapiens. Accession number, the same number that is assigned to this protein. After the accession, there is a version. This number version, the same accession number is present, and after that there is a dot and one is written here. This is the version 1. This accession numbers remains constant, but if the version number is changed from 1 to 2 or 3, it means the sequence itself is updated. Now it means it means that the sequence is not changed. It remains the same after submitting. Next is source. Here you can see the accession number of nucleotide sequence. If you click on this, you will directly go to the nucleotide sequence or the sequence, original nucleotide sequence. This shows the relationship actually, that the DNA sequence, this DNA sequence is making the messenger RNA with transcription, and then with translation the protein BRCA1 will be produced. So this protein sequence was translated from this nucleotide record. If you click on this, you will directly go to nucleotide record. Look, this is the nucleotide record. You can directly download the gene sequence of this protein. FASTA. This is the gene sequence of this protein. You can directly download this gene sequence. Organism, it's an Homo sapien. The sample is also taken from Homo sapien. Next is the uh, under the organism section, there is the taxonomic classification. It is provided. This is a phyletic lineage starting from eukaryota. Information indicates the evolutionary classification of humans. After that is a reference section. It actually provides information about the scientific publication. Here indicates the residues 1 to 18 1863. It indicates the number of amino acids from first amino acid, second, and then continue onward 1863 amino acid. These are the author's name who worked on this sequence on this protein. Next is title. It's a complete genomic sequence and analysis of 117 kb of human DNA containing gene BRCA1. This means that the researchers analyzed the uh, uh, 117 kilobase region of human DNA which contains BRCA1 gene. Next is journal. The journal name is Genome, and after that is it mentioned that this article is published in 1996, and the volume and the page number is mentioned here. Next is PubMed. PubMed number is given here. This is a unique PubMed identification number for research articles related to this protein BRCA1. So if you click on this, you will directly go to the research articles. Here you can see this comment section. This comment section actually describes very important correction in this protein annotation. Originally, scientists incorrectly identified this and then they corrected it. The incorrection was of exon. Uh, they discovered that this sequence was not a true exon after that. That's why they removed exon 4. Here you can see that 1A, 1B, 2, 3. The 4 is removed because it was identified as it is an incorrection. That's why it is removed. Next is feature section. In the feature section, you can get information about functional regions of protein. This is protein. This is the source. Here you can see the map of gene where you can map of this protein where you can find the gene of this protein. Here 17q21, it means 17 chromosome, q long arm, and 21 is the region in a long arm. Product is BRCA1, and protein is how much amino acids long? One residue to continuous, and then 1863 residues. So total residues in this proteins are these amino acids. Next is region. This region represents the ring finger domain. Here is a ring finger domain from starting amino acid 7 to 99. Here it actually this ring HC, you can see ring HC BRCA1. This is actually involved in protein interaction, ubiquitination ligase activity, and also in regulation of protein function. Here, if you open this, you can get the information about this. Here you can see the ring HCBRCA1. This is the zinc binding site. Here is a heterodimer. Going back, here you can see the site information of this protein. You can see there is a heterodimer interfere polypeptide binding, and also the serine rich domain associated with this protein, and also here you can see the first BRCT domain of breast cancer type 1 susceptibility protein and similar proteins. So here in this section, in feature section, you can get information about the functions of this protein. We can observe by going to the research sites of this, that what is the conserved amino acids, what are the variable regions, And where are the highly conserved amino acids? Suppose we are now opening this one. Let's study this in detail here. This sequence is conserved feature site is ACC1. This is the interaction site, and it's a polypeptide binding site. You can see here the sequence cluster subfamily hierarchy. NCBI also provides classification information about the protein. Here you can see the classification information. The sequence cluster, it actually groups different proteins with similar sequences into same category. Here you can see also the subfamily, where the subfamily of these proteins are also clustered here. It shows the relationship from general to specific. For example, which protein domain. We studied first protein BRCA1, then we studied the ring domain, then ring HC domain, then we studied now the protein structure. For example, we want to study the structure of this protein, specifically binding site. We can see here the structure, and then we will open this structure. This is the sequence alignment option. Here, these indicates that this sequence CCC is conserved. Next, CCC is conserved. Position and sequence is conserved in all species, like in humans, in elephant, in sea animals, in clonal radar. In these animals, this sequence is conserved. So here, if you want to get information about sequence alignment, you can get here the information. So we want to study the structure of BRCA1 protein. Here you can also see the protein structure. This is a structured database. Here you can see the structure. Here you can see explore the structure database. Structure database, it contains experimentally determined three-dimensional structure of proteins and nucleic acids. Here you can see this is the 3D structure or three-dimensional structure of proteins. It is very important to understand that the structure database is separate from the protein database. Before that, we are on the protein database, but now we are on the structure database. But protein records may be linked with this structure database. Protein sequence, it tells us about the amino acids. Here you can get information about the in amino acid structure. If you want to download the picture of this, you can also download here. Just select PNG and download the image. Now there is a main question that how to download the FASTA sequence of this protein BRCA1. If you want to download the FASTA sequence of this BRCA1 protein, then you directly go to the main page of this protein. Here you can see there is a written of FASTA. You can select this FASTA, and the sequence of FASTA protein. The sequence of protein will be shown to you. You can directly copy this information, or you can download this, copy this and paste in a notepad and then use this information, or you can directly download this file also. In today's video we learned about protein identification, references, sequence alignment, protein structure, and how to download the FASTA sequence of protein. If you found this lecture helpful, don't forget to like this video, subscribe to my channel, and share it with your friends. And thank you for watching. I will see you in our next lecture. In our upcoming video, we will continue this NCBI Bioinformatics series.